thank you everybody for coming to our session on 503Bs for quality validation and design. We have as our panelists Thomas Kupiec, Syed Salim, and Vicky Caldero, and they all have given some presentations that I'm assuming everybody watched at least 3 times and wrote down numerous questions. Amy Summers from Restore Health is going to be our moderator. She's also got a series of great questions for the panelists based on their presentations. And Amy, I'm going to let you take over from here.
Okay, thank you, Vivian. Everyone, thank you for joining us today. I know it is, taking time out of your day to learn and continue to grow is really important. And so I'm really happy to be here. And I'm not sure if I introduced myself yet, but I'm Amy Summers, and I'm a consultant to 503As and 503Bs. I am a pharmacist. I've spent my entire career in compounding, so it's my passion, it's my thing. And I had a chance last night to listen to all of the 3 presentations that we're discussing today. So I came up with some questions, and I hope at least it's a good start. And if you guys want to chime in with anything and be part of the discussion, I think that would be a great way to learn. The first question that I had was to each of you guys, where do you see the 503B industry headed from a quality perspective? Does it mirror commercial drug manufacturing? Is it more like a hybrid of manufacturer meets compounder? And also, do you think it's difficult to make all the entities happy? Vicki, do you want to start?
I'll take the last question of the first set of questions. And I think you said, is it difficult to make all the entities happy? Anytime you use the word all or never, the answer is yes. I mean, you can't please everyone all the time. So I think that question is, you're not going to make everyone happy, nor should you be trying to make everyone happy, even more so important. I think the bottom line is this quality. That's what this compounding pharmacy Grand Salon is about. And you want to have that quality, and it needs to be based upon scientific principles. And so I think I do see it moving. I think that the agency came out with some good guidance document on that 503B. I mean, even from the BUD, when we talk about stabilities and the numbers, they give you some ability to do 1 batch versus 3, calling it a limited stability. So I think there's some area that they're allowing for that hybrid. That's just in short my position or my opinion on those questions.
Sure, this is Syed Salim. I'm sorry, I apologize. I don't know why my camera is not working, but to address the question, so 503B is basically created to fill the gap, basically to fulfill the gap of shortage drugs, which not the big pharma is picking up. So, and to facilitate the necessary drugs to be made available at the same time, meeting the GMP quality. So the bar has been increased to make the compounded drug to go up to the level and quality of the manufacturing. But at the same time, as Tom was saying, there is a limited stability testing provision given. So 1, quality is maintained. Second, to fulfill the gap or need existing between the manufacturing versus a big compounder.
How about you, Vicky? Do you want to weigh in? I'm sorry? I can't hear you so well. It's very faint.
I wasn't able to hear. I didn't have... Okay, let's move on. Shit. All right, let's move on. I'll try to find out what's going on with Vicky. Amy, you just want to go with your next question?
Do you want to troubleshoot this?
Yeah, I'll troubleshoot this. You start with the next question.
Sure. Actually, Thomas, I couldn't agree more. I was sort of leading you to There's no way we're going to make everyone happy. So I guess a follow-up to your answer, how would you go about when you have an issue where maybe even the FDA might be easier in some cases to make happy than certain state boards of pharmacy?
That's weird.
Do you have any advice on that?
On the phone or, on the computer? All right, let's go back.
All happy. It's, by the way it's asked, I guess, from an emotional perspective, it's not about, like I said, making people happy. You want to do the right thing, whether they're happy or they're sad. You can't change a person's feelings, I don't think. I think ultimately it is our responsibility as a compounding pharmacist, 503A, 503B, or a commercial manufacturer doing a new drug application, investigating drug application, or abbreviated new drug application, a generic drug.
Yes.
It is our responsibility to put a safe and effective product out there. And as a compounder, you want to have the systems and processes in place from a quality. And I think that if you present your case, and I know we're involved in many instances like this, but if you present your case based upon the scientific data, based upon the guidelines that are put out by USP, FDA, ICH, I think they'll look at it, and it just needs to be presented versus have an agenda going into it with looking at it from a lens. Because, you may be wrong. I may be wrong in my assumptions, So I think we need to look at it more holistic. And I love this idea how Vivian has put this compounding pharmacy Grand Salon piece together, a collaborative approach. It's more of a question and answer, collaboration of all the speakers, coming together. So I think today, to be competitive, we need to be more collaborative in this compounding community. So I think that it's not about being happy. It's what is the right thing to do based upon the science and the guidelines. And, I talk about science and compliance a lot. And I think that's the idea, is bringing those two together.
Yeah, I really like that. So Vicki, it sounds like you're here with us. Yes?
I think I'm here. Can you
hear
me? Hi.
Do you want to add to any of this that we've been discussing?
What was the start? Because you're talking about stabilities, and I was getting a little excited, like, oh, I'm going to miss out on this.
Well, it was just a general overarching question about, where do you see this industry, the 503B industry, headed from a quality perspective? And is it more like a commercial drug manufacturer? Is it a hybrid, manufacturer meets compounder?
I think a little bit of both. I think the 503Bs are definitely going to be used to fulfill a need. And I think that need is speed. What I have seen is that during the hurricanes, they called on 503Bs. Are you going to be able to pick it up because there are drug shortages and, big pharma couldn't get things done so quickly to get to local hospitals. So I think definitely that is going to be something. I don't think it's going to be a small pharma company to wear stock on the shelves. I still think it's going to be something like that. But as far as quality, with all that being said, I don't believe that there's going to be any type of leniency. There's still going to be a level of expectation that is going to be, I like what you said, Thomas, about compliance with the science. So it has to be sound science, what's being put out there. So my experience with stability studies, yes, do a full study to get that dating on the product. And a lot of time people have a hard time because they are smaller, and that means making batches and not using them. It's solely for test, but you have to go to that level as if you were going to, be able to release the product. So that is kind of hard. That's tricky. And I think collaborations on how people have gotten around that and worked through that is great. But it's not to say that I think that it would be forgiven, as if you don't have to do that.
Yes, thank you for that. I feel the same way. I really like Thomas's science versus compliance. I really like that term. I was listening to it last night, and I that, yes, science versus compliance. And you said ideally these two systems overlap, but it's not always that way. There may be personal agendas. I'm just sort of paraphrasing here. But, we should make decisions based on quality and science. So do you have any examples within the compounding industry where science and compliance are not really working hand in hand at this time? Does anyone have any, maybe not just Thomas, but pretty much everyone open to answer this question? You want to start, Thomas?
From a testing perspective, it should be based upon the science. And from a formulation, if you're making a new compound, you need to understand the pharmaceutics, the kinetics, those aspects, if it's an extended release, the dissolution profile. So a lot of the formulation aspect of a compounding should be thought through and done on the science. So that's what I meant by basic should be the science. And then the compliance, of course, we want to meet the regulatory body. So if we're a 503B, we need to meet the guidance documents from the 503B that the agency has put out. Your 503A, you're probably looking more at the 795, 797, those issues. So I think that's what I'm talking about from the regulatory. In an ideal world, definitely the two together would come. And, it's probably not a perfect world, as we know it's not perfect, but you want to try to strive for the best of both, if possible, the greatest good for the greatest number of people, if possible.
I think from my perspective,
without sacrificing quality.
Right. Yeah, I thought, obviously science would help us all here. And I think that it gets a little tricky when compliance, if we're talking regulations, and then maybe there's room for interpretation. So you're thinking that your science is sound and it's bulletproof, but it becomes maybe interpretation of compliance with those regulations or guidances. So does anyone have anything to add on this?
So talking about the science and compliance, like Tom was saying, one example is the 797, proposed 797 versus CGMP. So there is a little difference in terms of requirements for those three currently. But again, compliance is the basic requirement, I would say. And then you build science upon that. So first meet the requirements, and then you build and add scientific judgment, justifications. I think that's probably a good approach.
Yeah, that sounds good. What do you guys think about 503Bs, and is there something that comes to mind that they struggle most with in their quality management system? Or maybe just in your own personal experience, what you've seen. I know, Syed, you talked about equipment validation. Is that a weak point for us 503Bs? Is it something else?
That is correct. But because the 503Bs are mostly coming from the compounding background, with the compounding history, now to catch up with the manufacturing regulations, the FDA, CFR, that is a little bit of a learning curve. Most of the times, compounding refers to 797 USPs. But when you get into the CGMP, you get with the CFRs, 21 CFR, Part 211, 210, and other Part 11 and all that. So that's the learning curve. And I personally have a little bit of a challenge in making the compounding personnel understand the CGMP regulations. So in a compounding world previously, you buy an equipment and you, based on the manufacturer specification, you just take the face value and you start producing it. You never had a chance to validate it or validate the equipment. Whereas when you come to manufacturing, you have to validate everything prior to producing any batches commercially. So yes, equipment validation is, again, talking about the quality management systems. Building the quality management system itself is something new to the compounding industry. So setting up a quality department, quality control, quality assurance, and how the microbiology aspects are taken care in terms of environmental monitoring, personal monitoring, cleaning validations, equipment validation, process validation, aseptic validations, APS, aseptic process simulations. So all these terms are fairly new to the historically compounding industry. This is a little bit of a learning curve. And again, I think the expectations of the FDA is to have the 503Bs, since they distribute the product, not dispense. So you fall into a different circle. And the expectations are, and the bar is up in the high there. So yes, the quality management system is a little challenging area, but again, I think we are evolving. 503B is fairly a 6-7 year old concept, 7-8 year olds, I think, 2013, so it makes about 7-8. So yes, that is a little bit of a challenging topic.
Would you say that 503Bs would be better off in getting some folks on board that have a GMP background?
Absolutely, yes. I agree. Absolutely, I agree. Having a GMP background definitely helps with team understanding the requirements, the need, and most of the times when you explain why and how the impact is, that makes it much of an easy to understand and follow. Yeah,
I'd say that's true. Vicki, anything to add to any struggles that you've seen with 503Bs and their quality management system?
A lot of what Saeed said, I agree with 100%. And also some of the 503Bs, again, they're smaller operations. So to have a dedicated quality staff, you have a lot of people who wear multiple hats in the beginnings as they start out and growing. So to have that arm's length of quality to actually be independent and removed from a production or manufacturing role is often sometimes a challenge. So I see that, and the difference between QA and QC, that quality assurance and quality control. Just thinking that QC testing is a QA, just to call that their QA, that's kind of hard. So it is something that that they're learning and struggling with, I think.
Yeah. Okay. Thank you for that. Since actually I want to ask you again, Vicki, I watched your presentation last night too, and I really liked what you were talking about. With knowledge comes great responsibility. I really liked what you said there. And I wanted to know if you could tell us of a time where maybe the knowledge that you gained, maybe the data you gathered or whatever it was, it made you into a tough spot where you were forced to make a decision that was really difficult. And I just wanted to know if you could share something that maybe had to result in something that was hard to do. Maybe you had to scrap a batch or initiate a recall or something like that. Is there something about that?
Well, I can bring up this time of an investigation that we were doing, and it involved a third-party lab for testing, and there was an OOS situation. So again, we have to do all investigations, and those get pretty involved because you have to do in-house to your facility, and then the product is shipped out and the handling, where it goes. So you're kind of doing an investigation in tandem with your testing laboratory. We came upon some information that there wasn't full transparency. So we did gain some knowledge of a root cause that unfortunately we had to make some tough decisions on.
Yeah.
Because it wasn't our data, it was their data that we had to get, and and it was a very tough situation, because how do we move forward from here? So ultimately, it broke the supplier agreement and the quality agreement in which we had in place with the third-party testing lab, which is another thing that I think 503Bs is to have those quality agreements with their suppliers. They can, and anybody that they're working with as their labs. A lot of times the labs now are very good that they have agreements that they send to the 503Bs, and I always advise, well, you should, have one as well. Any consultant you're working with, anybody, you should have a quality agreement in place. So, and if there is something like that that is broken, then you can base a decision on that. So we had a tough decision to, sever ties with that. That was an unfortunate one, but it was the lack of transparency. And as far as risk assessment was concerned, to move forward, we are using the information that is provided to us to make a decision on a release of product. And if there's not that, then the risk was too great to continue on. So, but when I make that comment of, with knowledge comes great responsibility, it could just be, it doesn't have to be something so catastrophic or drastic. It's just, if you find an issue, then what are you going to do about it? And I was having a meeting today, and I said, the one thing nobody has is time. Nobody has time. I get it. Everybody's busy. Nobody has time. But what you do have are priorities. So you just need to prioritize the work, and risk assessment comes in. So you can't just say, oh, I'll get to it later, or put it aside, and we'll just deal with it. If you know that there's something that might seem minor now, if you continue to let that grow, it's going to be a bigger problem. So put a plan in place with something. You've identified this. Now you have to do something about it, not worry about it later.
Yeah. Thank you for that. I actually had a question already outlined for Syed, and it has to do with disqualifying a critical supplier. So my question to you is, what do you do if you need to disqualify a critical supplier? Perhaps maybe you've learned that they have a concerning 483 or a warning letter, or maybe like in Vicky's case, maybe there was a product failure that you found the root cause to be that supplier. So I guess, what do you do, Syed?
Sure. Obviously, you have to have a due diligence and a constant watch on the supplier's behavior in terms of watching the FDA warning letters, and also getting the suppliers any issues he has in the news and such. So if you find a supplier, and if you, again, if it's a critical supplier, you don't have any other supplier who is able to supply the particular raw material. In this case, again, quality is number one. We go with the concept of the basic three principles: safety, efficacy, and quality. So if I'm doubtful of making any of these three, so in that case, that's the risk assessment you have to make. Is this product, or is it API which we received from this particular supplier, is safe, effective, and of the required quality? If this is not meeting these three parameters, that's the decision the quality would make in terms of
what's the far-reaching effect that it has? Do you need to do a stability study again or some other kind of validation if you had to disqualify your critical supplier and it was like your API, for example?
Absolutely. I think they have to go back to the books, do a risk evaluation impact. The risk evaluation impact is the critical thing which quality should take. Whatever the API or the component which is affected from that particular supplier is in question, they definitely need to go back and revise those batch records for any of the potency failures, any of the risk associated with the product, and then categorize into critical, major, and minor. Based on that, they would make a call, either including up to, in cases up to a recall, which nobody likes it. But if it is that critical, then up to that, you may have to take that step. And also keep this particular supplier on the watch list. I don't know if for any other products or component affected from this particular supplier. Not only that product which is affected, but also other supplied material becomes in question now. So again, it's a risk assessment and impact. These two terms quality should be looking into every single time.
Yeah, I agree. I want to switch gears and come back to Thomas a bit. I really enjoyed your presentation, and I know. Yeah, thanks. So I know that you mentioned your talk was to both 503As and 503Bs, and I think you did a good job at delineating some of that. You mentioned that evaluating 3 batches instead of 1 batch in the study is one of the difference points for study design for 503Bs versus 503As. So are there any other major differences from a 503B stability study design versus 503A, and even like the documentation of that?
Sure. I think the FDA has an allowance for 1 lot stability for 503B. I think they call this a limited stability. This shows that they know it's not a full stability study. And you're asking what other differences might be. I think the major difference of a 503B studies must be a protocol driven. So I think a protocol is the mechanism used to make sure that both the laboratory and the pharmacy are on the same page. The roles, the responsibilities are defined. But the method validation, the stability itself, it has to be a stability indicating method. So whether you're doing a stability indicating method for a 503A, a 503B, or big pharma, it should have the key components of the stressing, the acid, base, heat, peroxide, and UV. It has to be stressed. That's been noted for many, many years and decades from both the FDA, ICH, and USP. I think the method validation parameters are the same, but the largest difference I would say might be the amount of documentation required to meet the CGMP. I think for the most part, there's likely to be more data from a CGMP work because of the increased documentation, and thus the increased documentation or the batch numbers is going to, and thus the more review is going to be more costly from a CGMP perspective that when you do a study for a big pharma versus a 503A or even a 503B, which is more GMP-like.
Yeah, that makes sense. And with that, do you have advice for 503Bs to work smarter and not harder to maybe design their study? Meaning, like, is there a way to design a study that maybe, I don't even know, but maybe it covers multiple compounds, or I don't know, maybe it can be, yeah, go ahead.
That's interesting because we get that question all the time.
Yeah.
How do you design that study? And while you're trying to, that's a conundrum. That can be a double-edged sword because while you're trying to do a study with multi-constituents, I mean, we know from the pain management cream that was going on where they put in many different drugs in the same formulation. And even there's a drug-drug interaction. So the bottom line is that BUD or stability or shelf life study needs to mimic the exact formula that's being given to that patient under the same container closure that's being used. So you don't want to conduct your study using a different container than what was being used in a real life study. I mean, in the real life process. Also, the storage conditions need to be the same. So it's important to mimic what's going on in real life to conduct that BUD study in the laboratory. So those need to be in alignment. Okay, now you asked about, is there a way to be smart about this? Let's say you have multiple concentrations. One might be is where you could bracket those, but you need to be thinking of that, so you just maybe do the highest and the lowest. But I have seen where on both instances, on low concentration, there's a failure or an error, and on the high concentration, there's a failure and an error. So you need to look at that, look at it critically, understand the science, be able to evaluate all aspects of that piece.
Yeah, I have more questions I want to come back to, but I also want to give others a chance to speak. So let's get one for Vicki. Let's see here. You said something at the end of your presentation. I really you were getting passionate about it. You said when the inspector comes to your facility, or you said your stranger comes to your house, and they start asking, well, how do you know? And then you said, well, because I know. Well, how do you know? And I like that passion. And I just wanted to know if you have an example of how you knew that your CAPA was going to be good, that it would pass this stranger coming into your house and asking you, meaning in other words, can you provide us tips again in how to perform and document these CAPAs at a 503B?
And it's funny because I'm going to use a little bit of reference of what Thomas is talking about, because anytime a stability study, I always do everything in triplicate. And it's, once doesn't count, 2, you got lucky, 3, okay, we got some data. So I apply that to everything, and even the CAPA. How do you know? So trending is a way to say, well, it didn't happen again, so I guess so. But just like we stress the product during a stability, try to break your system, put it to the test. It's back to an experiment 101, test your hypothesis. Did it work? So, and doing that, putting those, a media fill challenge. It's a challenge. You're to put in worst case scenarios. So anything like that. And then you can say, well, we had a good run. We even tried, we tried something. We tried to simulate a problem or, an excursion of some sort. And that's how you know. And you're solid to that. And again, part of quality assurance is to assure that, you are in control, and that's being ahead of the game. So you're always looking for that foresight. So what's the immediate problem in front of you, you have to take care of that, but you're always looking globally and looking ahead. Like, how can we assure that we have this under control? So that's really where I said, do your due diligence to ask your whys. That way somebody else doesn't have to. Yeah. And again, for your house, your environment, this falls back to the compliance and to the regs. Everybody's facility is a little bit different. So you have to see how they work in your space.
This is really helpful. Thank you. I'm going to go back to Syed about, do you have any, you mentioned a minute ago about when you're using a supplier that has no competitor, like this is the only place you can get this item. So as a 503B that's kind of stuck with just one option, how do you qualify someone where, there's only one choice? And what would you do if it really didn't, meet all of your wish lists? Like maybe their supply chain's sluggish right now or something. Can you just speak on this, Syed, a little bit?
Absolutely. Again, it's not. I would probably would say the quality would need to look into the three basic principles: safety, efficacy, and quality. Our goal is to provide safe, effective product. Um, if at all the product or the API, the component is available, then we'll be able to produce one. If there isn't a product which is available, which meets the quality standards, then it doesn't make sense to make the product and take a risk. So providing the safe and effective product should be a priority. It's not the supplier. If the supplier is, if we cannot qualify the supplier, or we're not confident enough about the product, about the chemical, about the API, then I think there isn't any going further, because if the quality is in question, then going forward it doesn't make sense.
That's actually what I wanted to hear. I wanted to understand if I got it, because in my head I thought, well, if all my needs aren't being met from a quality perspective, then how can I move on? So I'm glad to hear you say that. Following up with another question to you, Syed.
Sure.
What should a 503B do if their supplier of an API changes the source of their ingredient? So let's say your 503B purchases an active from some supplier, and then their supplier, from manufacturer A has now been changed to manufacturer B. And pursuant to your quality agreement, they let you know what happened. But what would you need to do from a 503B perspective? Do you need to do anything on that?
Absolutely. There's something called pedigree. Pedigree means you need to go back to the source of the manufacturing. So the person who originally made the product. Now, as part of the quality due diligence would be to vet that particular manufacturer and the supply chain and everybody in between. There is information available to look up online, FDA registered manufacturers, the history of the original manufacturer. Partly the supplier who's supplying you the product has a obligation to qualify his own supplier. And when we are qualifying our supplier, we need to make sure and ask questions as to how he is qualifying his supplier. So if their qualification requirements are stringent, that basically make sure that they have a good and effective supplier qualification in place. One is to check that from your supplier, and also to vet the supplier who is now the new supplier, B. How long is he in the market? I mean, is there any kind of a FDA warning letters or any kind of red flags you can find for him? So basically, the quality need to do a full due diligence when they get these letters of change from the supplier.
Mm-hmm. Thomas, do you have anything to add in regards to maybe stability studies in this kind of example, where the manufacturer of your supply of maybe the API has now been changed?
Well, I know that is a concern from the agency. I think that individuals look at that process. Chemically, you want to ensure that the purity, the strength, the adulterant, there's no chromatographic impurity issues or residual solvents. I think one question you came up earlier, this is particularly of interest when you change the supplier of the API. Mm-hmm. That manufacturer of that API, you want to make sure it's consistent with quality, so it's not changing your product that you're giving to that patient. So whatever you need to do to ensure that and document that and show that at your level of regulatory authority, area, then you need to do so.
Yeah, that makes sense. Another one to you, Thomas. You were talking earlier in your presentation about, you sort of had a table, I believe, where you laid out the testing of sterility, endotoxins, and antimicrobial effectiveness. You had some at day 0 and maybe at the end point, and then some maybe had an intermediate time point. I guess it kind of goes back to my question about being working smarter, not harder. Maybe not saying doing the minimum to get the preferred result, but Like, for example, is container closure integrity, we only have to test it at the end. And then is that the only thing you have to do for container closure integrity? But then the sterility, I think you said ×0, and then maybe it was 90 and 180. And then I think it was endotoxin was day 0 and 180, and so was your antimicrobial effectiveness. So I just was wanting to hear a little bit more about those differences.
Okay, well, I think you're asking perhaps combined several questions. Like, what about container closure integrity for stability study of a 503B? Is the end of the good study good enough, or is day 0 is good? You know, I think the end is likely or potentially good enough, but again, more testing adds more power to your data. That adds the power of that. And you were wanting, like when you're looking at statistical probability or the p-value of it, when you're looking at a clinical trial or something, you want as much confidence in that it is the case. So it's important here as a 503B not to typically perform. I mean, typically 503Bs may not perform a container closure integrity testing as part of a batch release, but you do want to do it as part of that BUD type study.
For sure.
So I think when you're looking at that now, looking at microbial effectiveness, the endotoxin, you know, just doing it from the beginning or the end, what happens if you do have a failure? I mean, I think a question might be, I think someone from the audience asked about a pH difference, and can you qualify or change that aspect of it? You need to be able to analyze it. I love the idea how Vicki talked about a triplicate. But Vicki, what was your comment? 1 is not enough, but 2 is there, and then 3 is
you just got lucky.
I mean, I just want to coin that. I want to patent that phrase. You know, that's a good approach to it. You know, and I think that's why the agency, that's why you want to look at a triplicate. You want to see, is it reproducible? So, of course, these are costly. So I think you're trying to look at that. So 1, as long as the data passes, you're fine if you just do it 1 or 2 points, you know. But part of the reason to do stability is to look at the trends. And as we talked about in the 503A questions, two time points might make a trend, but it's a very weak trend. And so more time points add more power to that data. And
what if you were to fail in the middle somewhere on something?
It's a problem. I mean, it definitely is an issue. And then you have to go back and you look, you do all the investigations, you OS. I mean, it's important to understand on the OS investigations. I think there was a question alluding to that earlier about the stringency and the difficulty and the time and the cooperation. It's very important that you do have as a laboratory transparency and communication. And thus, again, echoing those quality agreements between the laboratory and the pharmacy or the 503B. It's the better the communication and open communication and transparency between the laboratory and the pharmacy, or the better outcomes I think everyone will have. Ultimately, the patient, which is what we're most concerned with.
Yeah, absolutely.
And can I add a little bit to this?
Yes, please.
The importance of that data and the stability study. I had an instance of an OOS that we had to do an investigation. And I was able to show by going back, I referred back to that stability data during that stress of the product to heat exposure. I was able to take that data and just put it right on top of the data that we got from the lab and say, how was the storage conditions? I mean, when it was received, it just so happened who was supposed to receive it was on vacation. It sat in an office. It wasn't refrigerated, but it was exact to what we had done during stability testing, and it wasn't an issue. So that data is so important to how your product's going to react. It's invaluable to have for the future, for if something does happen, that it's okay. We've stressed it for a poor condition, and here it acted exactly how it was supposed to, how we knew it was going to. So it made that investigation very clean, and I had solid evidence on the why. Well, how do you know? Well, you know why? Because I've tested it before. I've put it through this condition.
You can back it up. You said something in your talk. It was something about the only thing that looks bad is no action. So going back with if 503B is struggling with something over and over, maybe not even stability testing or anything like that. But let's say they keep getting a recurring out of specification of something, or they keep they keep having to have this deviation or incident or something. Do you, I guess, have advice for this kind of entity? Because you can't, not do anything. So what would you advise?
I would advise stop doing whatever it is and go back to the drawing board. And really look at that process, because unfortunately, if you continue on and don't do anything about it, but it's systematic, the only thing that is constant is the problem, then you just ticked up on that risk factor. And that's a dangerous place to be. You just need to stop, go back, look at the process, and reevaluate.
I had another question for you, Vicki. I think it's important, and I'm curious what your answer is. But, you said, don't worry, just write everything down. Just write it all out. So you've answered all the 5 whys, so they're not going to ask you and all that. So it's there. And so we can basically understand how you're going to correct and prevent and all of that. How would you handle a situation where maybe a regulator is reading your CAPA report or whatever, deviation log or something, and they're almost like they're trying to use these exhibits against you to find something because maybe they don't quite understand GMP fully? And I'm maybe not speaking about FDA, but just other sort of regulatory bodies that are new to regulating 503Bs. And so I think they're just, I guess just my question is how would you handle something like that where you've given everything, you've opened yourself up, and now it appears like this regulatory body is sort of trying to look for something to cite you on, any imperfection?
I think that's gotten a lot better because they're using SMEs. I know that in the beginning that was a big argument, is to have inspectors come in that weren't fully comfortable or
versed.
But versed, that's a good word. So in documenting everything, it has to be logical. Go through the steps of the story, and you did this, and this was the result. So it's resulting, and a lot is to show those resultings, all this information and everything, and all this work and investigation that you did. What did that yield? Which is why I spoke on if you can have things in quantifiable form, like what, show some hard data and some results. And if it's just process driven, then observations again are good. And then just have it all logically documented so that there doesn't have to be so much explaining. Like you said, we like during inspection to where we can hand over the documents, and like now they're doing a lot of things virtual or just asking for the documents ahead of time. They're not sitting across the table from us so much. And it has to make sense. So give it to somebody that's not their department. They understand and can read it and say, oh, okay, I understand how this happened. Or back up your actions, document what you did, and refer to, we followed process, and list that SOP. Cite yourself and say, this is what we did, so that's where they can refer to if they have any questions.
That's what I was hoping you would say, that, because you've opened yourself and been so confident in your remediations, then there's nothing really to say, I think. And I'm glad to hear that maybe whatever had been early on is now a little bit different now. So I think that's good to hear. I think I have a couple more questions for Saeed. You mentioned critical versus non-critical raw materials. What's the difference? Can you give a few examples? Are you there? Maybe you stepped away. Oh, there. Are you there?
Hello, can you hear me?
Yes.
Oh, sorry. Okay, so the critical raw material is anything, example being APIs, anything that has a direct impact and is able to change the pharmacology.
Okay.
So, again, anything which can which is going to affect the quality and safety of the product.
Okay.
That's basically the critical. And non-critical is anything which is directly not affecting these two parameters primarily.
So where does, like, if you're aseptic processing and not terminal sterilization, would the filter be considered critical?
Absolutely, which is having a direct impact on the product quality. Any API, chemical, packaging, any of these which is having a direct impact on the quality of the product is considered critical. So aseptic processing, filter, your personnel monitoring, environmental monitoring, API used, component used, packaging used. I mean, we're talking about leachables, making sure that the product doesn't have any leaching properties into the chemical. Anything directly touching the product would be critical.
And you recommended requalifying those suppliers that are like for your APIs to do it annually, just based off the guidance, and the other components every 2 years, and maybe in between if there's some changes. I guess I just wanted to infer that would that include other non-drug components like the filter or something like that?
Yes. APIs are definitely, as per the guidance, you have to qualify annually, at least minimum, and again, as risk-based. So for that reason, the quality needs to be keeping a constant eye on the supplier, or the chemical, the supplier, making sure that they are getting the quality product in. And talking about the filters, of course, definitely the filter is being one of the critical and most important component which is affecting directly the product quality. This definitely is, I would consider it part of an API. It would be like an API, as critical as an API. So whoever the supplier is, making sure that the filters are, what the filters we're getting is of quality, and whatever the claim they make. For example, PTFE filters and other hydrophilic or hydrophobic filters used for particular terminal sterilization, they need to be evaluated for the purpose what they're used. And also the supplier definitely need to be evaluated, and the manufacturer also needs to be looked for their specifications and again, the whole qualification process.
Thank you. We have 2 minutes left, and I wanted to ask one last question to Thomas. You had a slide on extractables and leachables, and I wanted to know if these parameters would be required for 503Bs to test as part of their stability study, or do they become more important when you're trying to assign a longer BUD? Can you speak on that?
Yeah, the extractable and leachables are not mentioned in the FDA draft guidance for the 503B stability studies, I don't believe. But they are important to consider from a safety perspective, as he is saying. If you cannot obtain the documentation from your vendor around those extractable leachables, you may wish to perform those studies. But we don't see many 503A or 503B clients requesting those at the present time.
Yeah, thank you for that.
That doesn't mean that they're not important, and so that you do need to obtain that data.
Yeah, I think it would become important for me just working it through in my head. The longer BUD you want to put on and the container closure system, the materials and all that, how it interacts. So I was just curious. I think we're almost out of time, Vivian. Is there any other questions that we needed to cover?
Nobody put anything, but now is the opportunity. If anybody wants to put anything in the chat or raise your hand. I mean, you covered so many great topics, Amy.
Thank you.
Yeah, does anybody else have anything they want to ask? Does not appear so.
Well, thank you guys for listening. I certainly had a good time, asking all these questions and learning from you guys. So thank you very much.
Thank you.
Thank you for the opportunity to work today with
Absolutely.
you guys. It's been a pleasure. Amy, Vicki, and Sia, it's been a pleasure.
Yes, thank you.
It's fun.
Yes, thank you so much. And this concludes today's portion of our live sessions. I hope everybody logs in and joins us tomorrow. Again, it's going to be 3 and 4 o'clock Eastern Standard Time, and tomorrow we'll be doing environmental monitoring again for 503As and 503Bs. And thank you, Amy and Thomas, for staying for both sessions, and Vicki and Syed. It was really great having you for this one.
Likewise, thank you.
All right, bye.